Excited to have the opportunity to share with you some recent work on using emerging AI hardware accelerator platforms to scale up evolutionary computation in a way that maintains our ability to observe what's going on inside of our evolutionary runs so that we can use that diagnostic telemetry to improve the performance of our evolutionary algorithms. So in this particular work, we used the Cerebras Wafer-Scale Engine, which is a really exciting emerging platform that puts 850,000 processor elements onto a single chip. Each of these processors operates independently and has its own local memory, and then they interface with each other in a grid lattice configuration, with each processor communicating with its neighbor through a messaging passing framework. So this is a really highly distributed architecture, which is indicative of a lot of the other really exciting AI accelerator architectures. Another example being the Cerebras the GraphCore IPU. And so on these really highly distributed architectures, the island model genetic algorithm makes a lot of sense as a way to map evolutionary computation onto the platforms. Under this model, you basically consider each processor as an independent subpopulation, and then you migrate genomes between processors at runtime. But this really highly distributed architecture introduces difficulty in collecting diagnostic telemetry. And in this particular work, we focus on looking at phylogenetic history, the structure of relatedness among genomes within the evolutionary run. Traditional approaches to collecting phylogenetic history and evolutionary computation use a centralized tracking model. There's various issues with scaling that up to these really highly distributed platforms. So instead, we use an inference-based reconstruction approach inspired by kind of the biological approach to studying phylogenies, where we annotate genomes in our simulation or in our evolutionary run with tag special hereditary stratigraphic annotation that's heritable. It gets passed down from generation to generation. And then we can do some comparisons among the tags held by genomes in the extant population and see how similar the tags are to each other. And from that, infer an estimate of the phylogenetic history of the simulation. In order to test this, we used a really simple, basically one max genetic algorithm under two treatments, one where we had beneficial mutations enabled and one where they were disabled. And then we tested whether we were able to detect the difference between these conditions through our reconstructed phylogenies. So these are two example phylogenies that we're able to reconstruct. Looking over several replicates, we applied phylogenetic metrics that we would expect to detect the difference in phylogenetic diversity, where we would expect greater phylogenetic diversity to be retained in the condition where we don't have beneficial mutations sweeping out the population. And we were able to successfully recover that signal from our telemetry. Working at the scale with this many processors introduces some really exciting possibilities to profoundly scale up evolutionary computation. We were able to run on, running on 500,000 processors, we were able to get a population size of 18 million running at 17,000 generations a second, which is enough to see about a quadrillion evaluations every hour. The phylogenetic tracking itself was lightweight. Annotations only require 96 bits per genome, and the performance cost was about equivalent to the rest of the minimalistic kind of toy example that we were using in this testing. So there's a lot more work to do with benchmarking and looking at some more robust scaling analysis and taking this hardware and applying it to more involved benchmarks and domain problems. There's a lot, I think, to look at about how to organize niching for multi-objective optimization onto this spatial environment. And we've designed the code that we used to be modular and easily extensible, and it's freely available on GitHub. So you can take this and use this platform for your own problems. With that, I'll acknowledge my co-authors and thank you for your time.